All right, Lab 7 review, microanatomy of the lymphatic system. This is for histology. Let's get started. All right, so we have the microanatomy of the lymphatic system, just a quick overview. Lymphatic system is going to carry lymph, circulate lymphocytes, return proteins, transport antigens to lymph vessels, and then transport lipids from the GI tract to the blood. And when we're looking at the vessels, it's thin-walled, continuous, no red blood cells. All right, then we're dealing with the cells that we're dealing with here. We have our lymphocytes, T and B lymphocytes, helper T, cytotoxic T, lymphocytes, B lymphocytes can take the form of plasma or memory B lymphocytes, but we'll also just see them as undifferentiated B lymphocytes here. We have our antigen-presenting cells, which are going to be our macrophages, basically the phagocytic cells that will take advantage of major histocompatibility proteins, the MHC2. They'll eat the antigens presented on the MHC2 to our helper T cells, which can then show those go activate cytotoxic T cells or activate our B cells to become plasma cells. All right, so that is great information, but what does that mean for histology? We're going to be looking at the lymph organs and then a little bit of the diffuse lymph tissue. The idea here is that below the epithelium of, say, the gut or mucosa or the lungs, we have diffuse lymph tissue, which is going to contain these immune cells. And basically the goal is any antigens that get through the epithelium are going to run into our diffuse lymph tissue where our immune cells can get to those antigens. All right, then we have our lymph organs. And so the idea is that the lymph vessels will take lymph to the lymph organs. And really what we have here are the primary lymph organs, which consist of the thymus and the bone marrow. And these are the places where T cells and B cells are going to differentiate from basically their precursor stem cell into the T cell and B cell. Right, and then after they have formed B cells or T cells in the thymus for T cell and bone marrow for B cell, they will go to the secondary lymph organs and they'll become immunocompetent after being exposed to specific antigens. All right, and so what are our secondary lymph organs? We have our lymph nodes, our spleen, our lymph nodules, also called lymph follicles. We have our tonsils, and then we have our appendix, and we're going to go through these for the lab today. All right, so starting with our diffuse lymphatic tissue, we have an example here. We can see that this is most likely probably GALT, so gut-associated lymphatic tissue. We can see that we have the epithelium here, and then deep to that, we have connective tissue, which is where our diffuse lymphatic tissue is going to be. And so this is consisting of our T and B lymphocytes, plasma cells, antigen-presenting cells. If you look at it really closely, right, you zoom in here, we have our, these are actually plasma cells. You can see kind of some clock face or spokes of the wheel in the chromatin pattern of the nucleus. But they are plasma cells, which are differentiated B cells. So big round nucleus, not too much cytoplasm, but they've differentiated further from the sort of generic lymphocyte into generic B lymphocyte into the plasma cell. And so you can see a little bit more going on with the nucleus than you could previously. All right, then we have our primary lymphatic nodules, and it's important to know there's no capsule here. You can see this is the nodule right there. It's just a bunch of congregated lymphocytes. If you were able to zoom in, you would see that they are just the standard lymphocyte. They're uniformly basophilic in terms of the nucleus, and they are therefore not activated. They're immature. They look like regular lymphocytes because they haven't undergone antigen-dependent proliferation. They're not plasma cells. These are just B lymphocytes, like we've seen in our previous lab 5. Okay, and so primary lymphocytes located typically just below epithelium, surrounded by glands and diffuse lymphatic tissue. Right, the idea is that our lymph vessels will get lymph to nodules or any of our secondary lymph organs in order for the antigens to be exposed to lymphocytes. But primary lymph nodules are inactive B lymphocytes. All right, then it's like, okay, well, what if you do get some exposure? We have our secondary lymph nodules, and you can see that you have these white specks inside of this germinal center. Right, this is the germinal center. And this letter color is from differentiating B cells. They're starting to get bigger. They have more cytoplasm, starting to become paler staining, and so they are becoming our plasma cells. And then outside of that, we have this darker region, and this is called the mantle zone or the corona. It's just more B cells, but they haven't reacted to an antigen, so they just look regular old lymphocytes. Okay, and there's lots of these secondary nodules in the ileum of the small intestines, and we would call them Peyer's patches. Lots of secondary lymph nodules, and primary lymph nodules. So Peyer's patches in the ileum of the small intestine. Again, nodules do not have a capsule over them. Okay, then we had tonsils. So tonsils are another secondary lymph organ. It's associated with, so different tonsils, not all of them are associated with the oral cavity, but the ones that we care about at this point are, the one that we've given in this example is the palatine one. It's really just a big mass of lymphatic tissue that has, this one does have a capsule, dense irregular connective tissue, and it'll have both primary and secondary lymph nodules, and you can kind of see that here. Here's a primary lymph nodule, again, B lymphocytes. Here's a secondary lymph nodule, B lymphocytes, and then outside of these nodules, in the space in between, you'll have T lymphocytes, right? And then in tonsils, you have tonsillar crypts, which are invaginations of the epithelium. This isn't the best picture, but this is one right here, and a better example would be one that's cutting through clearly from the epithelium, cutting downwards, right? But tonsillar crypts, it's not a trabeculum, which we'll see later in our lymph nodes right here. So this is a lymph node, also a secondary lymphatic organ. Its function is to filter lymph. It does have an invagination right here of the capsule downwards, and we call this the trabeculae or trabeculum, and it's from dense regular tissue of the capsule. Then we have our parenchyma, which is the name for the cellular elements of an organ. the breakdown of the lymph node is we have in this region A out here, we have our cortex. Then in the region B, we have our paracortex. And then, or excuse me, region C, we have our paracortex. And then B right here is our medulla with dark areas, which are our medullary cords, and light areas, which are our medullary sinuses, which are vessels. Our cortex is considered the B-dependent zone. As you can see, we have secondary nodules, we have primary nodules. And so the cortex is B-dependent zone. Our paracortex, so you can see here, this other zone that does not have these nodules are going to be the densely packed T cells, and you can find them partially based on it's going to have these specialized blood vessels, HEVs, in them, which is a good way to tell. And then the medulla is very easy; it's the deepest layer. It's going to have these different colors, and then in particular, it's going to have the hilus in it, which is going to contain a artery and a vein. All right, in terms of flow of lymph into a lymph node, we have that invagination, the trabeculum of the sinus inwards. So lymph is going to go into the subscapular, right, so the capsule sinus, and then it will go from there into the trabeculum. The sinus in particular that it's going into is continuous on the side of the connective tissue, and it's discontinuous on the side of the parenchyma. And so the idea is that the lymph going into the sinus is going to be able to flow through the cortex and the parenchyma there. And so, hopefully then your B lymphocytes can be activated, become plasma cells, and we're filtering lymph. All right. So sinuses, as we said, are continuous when adjacent to connective tissue, discontinuous when adjacent to parenchyma. Okay, then we go to the medullary part, which is the very inner part. We said that we have the hilus in this region, and so the hilus is right here, where we have a vein right there. We have a muscular artery, and we can see that we have muscular arteries. We remember have 3 to 40 smooth muscle layers, and so ideally we could count that, but it's pretty low. Can't see too much detail in this picture. And then 3, we have the medullary sinus. It's discontinuous since it's bordered by parenchyma on all sides. Then we have our cords, which have reticular cells and help organize cells into cords. And then finally, again, we have our hilus, which is where lymph can leave via the efferent lymph nodule or vessel. All right, and then blood flow through the lymph node. If we take a look at the paracortex, we have a muscular artery right here. It will branch off and give capillaries, and then on the other side we have a capillary venule. So the HEV, it has simple cuboidal or simple columnar epithelium, and it's going to be the site where lymphocytes can leave the blood to enter into the lymph node, right? But they can't exit via the HEV. They can only enter. They can exit at the efferent lymphatic vessel at the hilus. All right, and then the silver staining of the lymph node. Basically, it's to show we have a lot of reticular fibers in our lymph node that are doing the organization, and you can see that in the cords, the darker regions, you have a lot of different lymphocytes, and you have more reticular fibers, and then in the sinuses, you have some reticular fibers, but again, it's mostly set to the cords. Okay, then we take a look at spleen. Again, another secondary lymph organ. Function of the spleen is slightly similar to kidney in the sense that it's going to filter blood, but the idea is you're filtering blood. Sorry, it says to remove macrocytes mix. You're filtering blood to get rid of basically old red blood cells. It also is going to store red blood cells and platelets. In terms of the structure, you have connective tissue capsule, and then the parenchyma is broken up into white pulp and red pulp. Okay, and so the white pulp are basically nodules that are oriented around an arteriole called the central arteriole. And so the idea is that there's going to be multiple of these. We can see one here, and that's what we're getting in this picture. But here we have a central arteriole, and this is going to be kind of like if you imagine you stick out your arm, that'd be like the central arteriole, which we call central artery. And then around that, if you're wearing a long sleeve shirt, that would be the Powell's region, which we call the periarterial lymphatic sheath, and that contains a bunch of T cells. All right, so this region around it is going to contain a bunch of T cells. And then we have the white pulp right here, which is our node, and this is a secondary lymph nodule, excuse me. And just like before, you have the germinal center with the developing B cells, and then you have the mantle or corona on the outside with the B cells that are inactive. They have been activated by an antigen. Okay, so here's another cross-section of the white pulp. This is a cross-section in the sense that we're cutting through the central artery. So here's the central artery. Then we have our pals. That's our t-shirt. This is our arm. This is our t-shirt over the arm. And then our nodule white pulp is like your fist at the end, or some sort of bump on the side. And so 2 is the germinal center, 4 is the central artery, 1 is the T cells, the pals, and then 3 is the mantle or corona of that secondary nodule. All right, the red pulp are the sinuses in the spleen, and so we call them red pulp. It's a little bit harder to tell here, but you do have a bunch of red blood cells, and which is why it's called red pulp, even though in this particular staining, you really can't see the red blood cells very well. But the red pulp are the sinuses. It's discontinuous capillaries. There's red blood cells in them, not to be mistaken with the sinuses that we saw in the lymph node, right? So if we take a look at these sinuses, let's see, right here, right, we had the medullary sinuses in the lymph node, these guys, and then these are the medullary cords. The medullary sinuses, you can see there's a bunch of cells with nuclei in them, and that's a good hint that that's medullary sinus versus here we have the spleen sinus, which has a bunch of red blood cells, but you can see that they're formed elements. We don't see any nuclei, right? So the goal, right, of the spleen is to filter blood, destroy old red blood cells, and then outside of the sinuses, we have the cords, and you can see that they do have the lymphatic cells in them, as we have these nice circular nuclei, with not very much cytoplasm. Again, it's hard to tell in this because you can't see too much detail, but if we take a look at the electron microscopy of a sinus, you can see that we have some discontinuous, essentially discontinuous vessel capillary, and then we have some red blood cells inside of them. All right, and then we have our thymus, which is our first primary lymph organ, and we have our T cells are going to differentiate to become T cells. Right before this, they were from the bone marrow. We got precursors, and they become T cells in the thymus. But before they get to the from the bone marrow, we get the precursors. They go to the thymus, and then they can differentiate in the thymus and become T cells. And so the thymus has got two parts, just similar as before. We have the stroma, the connective tissue, which divides the thymus into lobes that we can see here. The thymus does not contain reticular fibers. It does have reticular cells that have these little processes that come out, but no reticular fibers. Right, so this shouldn't have any reticular fibers. And then we have a cortex and a medulla for the parenchymal elements. All right, so cortex versus medulla. The cortex is going to be where our basically the maturing T cells are going to be, and then our medulla is going to be where our mature T cells are. Okay, so they go from cortex to medulla, and then how can you tell medulla versus cortex? Well, you can see a little bit of difference in the staining here, but the big one is these guys right here, which are the Hazel's corpuscle, and we were told that these are epithelial reticular cells of some sort. We don't really know what the function is at this point. They have some sort of function, but we just don't know what they are, just what we were told in class. And then another thing to know, the tiny little round cells we see in the cortex, those are our thrombocytes, are T cells that are maturing. Okay, important to know that the capillaries that we see throughout are continuous to ensure the blood-thymus barrier, which is to protect our thymocytes as our T cells are maturing. Okay, and where do you want over this? Mature T cells are, okay, yep, mature T cells leave the thymus through the medulla. And great, so let's do a quick review. We have our lymph node, we have the cortex, which is B-dependent, so that's one, and then we have our paracortex, and then we have our medulla with the sinuses, and we know that, let's not put, this is 1, this is B-cell dependent, this is T-cell dependent. Okay, so 2 is our T-cell dependent region, and then our medulla has the medulla sinuses. All right, so 1 is medullary cord. Why we know it's dark. 2 is medullary sinus. All right, where in the lymph node is this? In the medulla. All right, blood flow through a lymph node. We see that we have blood is coming in through our artery, and then it's going to leave through our vein, right, just like this. And then, of course, we have here the opportunity for, we have our, right here, our HEV, which is where our lymphatic cells can enter into the lymph node. And then in terms of where that is on this guy, we have it right here. You can see, if you were to try to draw the edge, the ring of the HEV, again, it's a venule, so it's not going to be completely circular, but it is simple cuboidal or columnar. All right, and so here's just another example of an HEV. There where T cells and B cells can enter through a process called diapedesis. On the lower right, we can see that HEV, just find another picture of this, our T cell is, or lymphocyte is going out of the HEV, so into the lymph node, right? So HEVs are one way, it's from blood into lymph node, so out of the HEV, from blood to HEV to lymph node, but not back into the blood. All right, so T cells stay in the paracortex, and B cells are going to go to the nodules in the cortex, so they move away. All right, then we have our spleen. Spleen is surrounded by a capsule made of dense regular connective tissue. Reticular connective tissue, remember we said in the thymus we don't have this, but spleen we certainly do. The red pulp is numbered 2, white pulp is numbered 1, right? White pulp, we have those nice white nodules, and they're oriented around central artery. All right, so red pulp of spleen, remember that we have those sinuses, and they have a ton of red blood cells in them. So look at these nice red blood cells. We seem to have discontinuous, right? Discontinuous sinus, which is also a good sign. So this is spleen red pulp. All right, spleen white pulp. Okay, so this is another situation. This is a cross section of white pulp. One is the germinal center of a secondary nodule. 2 is going to be the mantle / corona, again, of a secondary nodule. 3 right here, that appears to be, that is not the best picture. If you zoom out a little bit, but splenic cords and sinuses, so this would be red pulp. And then 4 is our central artery, which we know, of course, is really in arterial. And then if that is our arm, then pals, which has our T cells, is like our sleeve, and then our secondary node is like our fist, or some sort of, maybe you have a gigantic tumor on the side of your arm. All right, blood flow through the spleen. We have the central artery with the splenic node, or secondary node around it. We have the pals as our sleeve. And so we have our sinuses that take in red blood cells. And yeah. All right, so that's it. That was the end of Lab 7 review. Good job, guys, and keep at it.